Good morning, students. In this video we are going to see about the gene expression. So it is a regulatory system that controls the genetic expression. So till now you have studied that the structure of DNA and how it is replicated and how it is transcribed. So this transcription or how the gene is expressed into its needed form should be controlled. So our body is in a homeostasis. So it needs certain products at certain time. Certain times it is not needed. And when there is a environmental sensitivity or when there is an environmental change, our body need to adapt to that. So for that adaptive response, we need the control of gene expression. So if we need something more genes should be expressed. If we need to control the production of something, less gene need to be expressed. So when we are studying certain metabolic pathways, in the regulatory step of most of the metabolic pathways you would have studied, there is a genetic control. So it is due to the expression of gene. So basically we have the gene, but we are controlling its expression to produce the desired amount of the product from the gene. So the most common regulation where we do is at the level of transcription. We can control at the DNA, but the most common regulation occurs through the transcriptional control. So the next thing is, for the cellular function, the gene expression determines the cell's phenotype. So for a particular type of cell, certain things are needed. Certain functions the cell will provide. For example, a cell present in the gland will produce certain hormones. So for that, it has the gene that determines its cellular phenotype. So this expression is regulated in our body to make the actual homeostasis in the body. Also, in order to respond dynamically to the internal and external stimuli. So when there could be some stimuli from the body to maintain the homeostasis, or some external stimuli like the changes in the environment which the body need to be adaptive. So for all these things, the gene expression plays a very important role, and the most common regulation where we do is at the level of transcription. So the next thing is, our body has two types of genes. One is housekeeping genes, other is inducible genes. So what is housekeeping genes? They are expressed constantly in all the cells for basic cellular functions. That is, to maintain our body's basic cellular functions, these genes are expressed constantly. So they are known as housekeeping genes. So example for that is the gene for essential metabolic pathway enzymes. So they are needed for a constant energy production in our body. So these genes are expressed constantly for that. The next other type is inducible genes. They are expressed only under certain special circumstances. That is, when a specific signal is given to them, these genes are induced to express, and they will be transcribed and then translated to protein. So these are the two types of genes present in our body. And next thing is, what is activator response? When the expression increases, so that is known as activator. So the activator are inducers. The inducers, they will increase the expression. They are known as activator. Whereas if you decrease the expression of the gene, then they are known as repressor. So things which are activating the or increasing the expression, they are known as activator. Things which are decreasing the response, that is genetic expression, they are known as repressor. So how we are going to regulate the gene? So we can do at various levels. The most common is at the transcriptional level, and the other thing is at the DNA level. So these are the two important ways of gene expression, regulating the gene expression. So the transcription level is, we can either induce or repress the transcription, or we can do the process the mRNA like alternate splicing or different types of RNA editing mechanisms. So these things will alter the or regulate the gene expression. Other thing is the mRNA stability. So we know that the post-transcriptional modification of mRNA is mainly to stabilize the mRNA. So by changing the stability, that is, if you want more gene to be expressed, we need to increase the stability. If you want less gene to be expressed, the stability should be reduced. So by altering the stability, gene expression can also be altered. And other thing is non-coding RNA. So in the RNA class, you would have studied about some RNA like small interfering RNA, siRNA or micro RNA. So these RNA, they will promote more mRNA for degradation, and thereby they will regulate the gene expression. If more mRNA is degraded, less amount of gene will be expressed, less amount of protein will be produced by translation. So these are the ways by which we can alter the gene regulation by at the level of transcription. Whereas we can do even at the level of DNA level. That is, we can amplify the gene, we can rearrange the gene, or we can do some transposition. So these changes occur in the body. Either the gene can be amplified, rearranged based on the need of the body. So these are the changes which are happening at the DNA level. That is at the DNA sequence level before the transcription. And other thing is epigenetic modification. That is, we can alter the chemical structure around the DNA. That is, we can methylate the DNA, we can do modifications in the histone, that is the protein surrounding the DNA. So this will alter the chromatin accessibility. So mainly we are regulating the gene at the two levels. One is at the transcription level, and other it is by the DNA level. So for understanding the regulation of gene at the transcription level, we can consider one model, that is lac operon structure. So this is occurring at the prokaryotes. So this is an example of gene expression in the prokaryotes. So what is lac operon? So it is a gene with these elements, that is, it has structural genes lac Z, Y, A. These genes encode for separate enzymes and proteins. So that is lac Z, it is responsible for producing beta galactosidase enzyme, and lac Y is for producing permease. So what is the function of beta galactosidase? It breaks the lactose into glucose and galactose. And permease, it is a carrier protein. It facilitates the lactose transport in the bacterial cells. So it is mainly seen in the prokaryotes. And other structural gene is lac-A. So it codes for thiogalactosidase, galactoside transacetylase, and the function of this is still unclear. So these are the three structural genes which are present in the lac operon structure. So in addition to that, the lac operon structure also has a regulatory element. That is, it has a promoter, it has an operator, it has a repressor. So we need these regulatory elements along with these structural genes. So basically a gene should express and produce its product. So this is what we need. But in addition to that, it has a regulatory element to control the production of these products. So we can see clearly in the following slides. So how the control is taking place? So first scenario, that is, as we seen already, the regulation of gene expression is based on some stimuli. Either it could be internal stimuli or external stimuli. So here the stimulus is based on the presence and absence of lactose and glucose. So the aim is the bacteria need glucose. So if glucose is absent, it utilizes lactose to produce glucose. So here the lac Z gene, it produces the enzyme beta galactosidase, which will produce glucose from lactose. So in the absence of glucose, this gene should be expressed. That is the main aim. So first, here we are seeing that this gene has these structural genetic elements, that is lac Z gene, lac Y gene, lac A gene. So these need to be transcribed. But before this genes position, here we can see certain other genes, that is lac I gene. It will produce repressors. That is, what is repressor? It will reduce the gene expression. Okay. So now the first scenario is no inducer. That means no lactose. Lactose is the inducer here. So here we have glucose, but lactose is absent. So what happens here? Basically, this repressor gene will produce repressors. So this repressor will bind to the operator region. So what happens? This RNA polymerase cannot bind to the promoter and no transcription is taking place. So there is no inducer, only repressors are here. So the repressor will stop the transcription process. So no transcription is taking place here. The next scenario is we have an inducer and we are having glucose. That is, glucose is present, lactose is also present. So what happens in this condition? So the inhibitor, the repressor gene is transcribed and repressor are produced. But since we have the inducer here, what is the inducer? That is lactose. Lactose is present. It will bind to the repressor and make it inactive. So no repressor is going to bind here and repress the process here. So the inducer is present here. It will bind to the repressor and it will inactivate the repressor. So what should happen? Ideally, the transcription should proceed. But in order to proceed the transcription, the RNA polymerase need another thing, that is CAP, catabolite active protein. So activator protein. So this CAP will bind with cyclic AMP to form a complex. So this complex is essential for the RNA polymerase to efficiently bind to the promoter. Since the glucose is present here, we have both glucose and lactose. So when glucose is low, this CAP, catabolite activator protein, will be increased and more this complex will be formed and this RNA polymerase will efficiently bind to the promoter. So but what is happening here? There is no repressor. The formed repressor is inactivated by the inducer, that is lactose has inactivated. The RNA polymerase can do the transcription, but due to the absence of this catabolite activator protein, that is produced only when the glucose is low. Since glucose is present here, this binding to the promoter is not taking place and no transcription is taking here. The next scenario, here lactose is present, glucose is absent. So you can imagine like this, the bacteria has lactose but no glucose. So glucose is very vital molecule which is needed for the survival of the bacteria. So now glucose is not present here. In the first two scenarios we have seen that in the first scenario glucose alone is present. The second scenario glucose and lactose are present, but here no glucose. So now the bacteria need to produce glucose from this lactose. So in this condition, the gene for producing beta galactosidase protein, that is which will produce glucose from this lactose, it should be transcribed. So what is happening here? There is inducer is present here, that is lactose is present here and no glucose. So what is happening here? The repressor is producing, but since the inducer is present here, it will inactivate the repressor. So no repression here. Since glucose is low, glucose is absent, there will be adequate amount of catabolite activator protein, that is CAP, which will form a complex with the cyclic AMP, and this complex promotes the binding of RNA polymerase with the promoter region and the transcription is started here. You can see the RNA polymerase is moved from here to start the transcription process and it has produced the respective proteins. Lac Z has produced beta galactosidase, lac Y permease and lac A this acetylase protein. So the needed gene is expressed here. That is a concept here. So when the in response to the stimuli, the needed gene is transcribed. So this is a basic concept to understand how even though the genes are present here, the expression of the genes is regulated based on the environmental or the stimuli which it is getting. So it will not be expressed always. When it is needed, it is expressed. So in order to understand that, we have considered this structure, that is lac operon concept, which is present in prokaryotes. So what we have seen in the lac operon regulation mechanism, when glucose is present, the repressors are active and they will block the RNA polymerase transcription. When lactose is present, it causes conformation change in the repressor and make them inactive repressor. So ultimately it will be in the de-repression state, the RNA polymerase transcribe the structural genes. So there is a term called gratuitous inducer. That means some lactose analogs like isopropyl thiogalactoside. So they can induce the lac operon structure without any metabolism. And the ultimate takeaway point is, even though we can see that glucose will activate the repressor, lactose will deactivate the repressor, the ultimate deciding point is glucose. The glucose is the priority point. So its presence overrides the lactose induction. That means even though lactose present and the inducer causes de-repression or change the conformation change of the repressor, we need the absence of glucose to produce the catabolite activator protein, because that is what determines the binding of RNA polymerase to the protein. So finally, to make it clear, when glucose is absent, the lac operon structure will become active. When glucose is present, irrespective of the presence of lactose, the lac operon structure will become switched off. It will become inactive. No transcription is taking place. So glucose is the final deciding factor in this regulation mechanism. So a few words about catabolite activator protein, that is CAP, present in the lac operon. So it is a positive regulator. So when it is present, it will activate the transcription. So what is the requirement for that? It needs low glucose. That is the stimuli needed for this protein to be activated. So when glucose concentration is low, there will be increased cyclic AMP levels, and the complex between these two will be formed in increased quantity, and that will promote binding of RNA polymerase. So there will be enhanced transcription and more gene will be transcribed. So when the repression will take place, when high glucose is present, the opposite is going to take place. Cyclic AMP will be low, and this catabolite activator protein cannot form this complex, and that will reduce the RNA polymerase binding to the promoter region. So next, coming to the regulatory mechanism by altering the stability of mRNA. So the mRNA, after forming, it will be processed in the post-transcription stage to become stable. So how it's what are the factors will affect the stability or alter the stability, whether increase or decrease the stability of this mRNA? The first thing is ribonuclear protein particles. So when the mRNA it goes to the cytoplasm, it will exist as ribonuclear protein particle. That is, it will bind with the protein to form this protein particle. So this ribonuclear protein particle RNP is a protective protein. So it increases the stability of the mRNA. So next thing is P bodies, that is processing bodies. They also serve as a major site for mRNA metabolism regulation. And other thing is, the produced mRNA should not be degraded or it is acted by the nucleus. So it should be protected from the nucleus. So the protein binding, this kind of binding of protein will protect the mRNA from degradation by this nuclease enzyme. And at the 3' end, the mRNA will have a stem loop structure. This also will prevent the exonuclease attack and regulate the message stability. And also the 3' end untranslated region containing AU sequence, adenine uracil sequences, also shorten the mRNA half-life. So if at the 3' end there is presence of more amount of this sequence, AU sequence, then that mRNA will become degraded soon. It will shorten its half-life. So here we have seen some things are increasing the half-life or increasing the protection or increasing the stability of the mRNA, whereas some are decreasing the stability of mRNA. So based on the need for its expression, we can decide whether the stability should be increased or stability should be decreased. So this is an area where we can regulate the mRNA stability, thereby we can regulate the gene expression. So till now we have seen about regulation at the level of transcription. So we have seen the repressor and inducers, that is by using the lac operon concept, and we have also seen the mRNA stability by changing the stability of the mRNA. So these are the certain mechanisms by which the gene regulation is taking place at the transcription level. Now we are moving to regulation at the level of gene, that is DNA level. At the DNA level, what are the regulatory mechanisms taking place to alter the expression of a particular gene? So first thing is gene amplification. That is, if we need some product to be produced more, we can amplify that gene in more numbers. So increasing gene copy will increase the transcription and thereby increasing the protein synthesis. So one example to understand this is, we have heard about the drug called methotrexate. What is it? It will inhibit the dihydrofolate reductase enzyme. So this drug is used for cancer treatment. So when we are treating that patient with this drug for a long time, it causes resistance to this drug. So how the resistance is taking place? The cancer cells will produce amplified genes for dihydrofolate reductase. That means we are targeting dihydrofolate reductase enzyme to be destroyed. It should be inhibited, but the cancer cells are producing more amount of dihydrofolate reductase. Even though we are targeting it with the drug, we are reducing it with the drug, it amplifies the genes needed for dihydrofolate reductase and produce more dihydrofolate reductase enzyme. So it causes methotrexate enzyme. This is an example for gene amplification. And other method is gene rearrangement. So it is seen in immunoglobulins. So we have different types of immunoglobulins, and the immunoglobulins have variable region, joining region, and constant segments. So each segment is produced from a certain gene. So by having a various combination of these genes, that means we have V is, that is variable region. So it has a separate gene, D has a separate gene, J, that is a joining region, has a separate gene. By taking different type of V, different type of D, different type of J, we can rearrange and produce different types of immunoglobulins. So which immunoglobulin is needed at that time is decided by the rearrangement which we are doing. So here we are controlling the gene expression at the DNA level by two methods. One is gene amplification. When there is a need, we are producing more number of genes, more copies of genes. Whereas other method is when a particular variant is needed, we are rearranging the genes to produce that variant in more number. So this picture shows how the gene amplification is taking place. So for example, gene A and gene B, that is, if you think that these are the genes which are needed for producing dihydrofolate reductase. Now the patient is taking methotrexate for a long time. So basically this gene A and gene B will be destroyed by the drug. But what the cancer cells does, it produces more amount of gene A and gene B by gene amplification. So the normal dose of our drug cannot eradicate this amount of this dihydrofolate reductase because it is bypassing or it is overgoing the amount of inhibition given by the drug. It will produce more amount of dihydrofolate reductase by gene amplification. So this is how the gene amplification takes place. Now the other method is gene rearrangement. So we have seen that for this immunoglobulin to be produced, it needs a variable region from this V2. It needs a constant region from this D1, and it needs a joining region from this, that is J3. So it produces a particular immunoglobulin protein, and this produces another immunoglobulin. This produces another immunoglobulin. So what is happening here? We are rearranging. This is the basic gene, and from this we are rearranging according to the need of that particular immunoglobulin. So if this immunoglobulin is needed more, we can rearrange this formula in more times. So when this rearrangement is taking place in more amount, more this type of immunoglobulin gene will be expressed, and that particular immunoglobulin will be produced at that time. So it is based on a particular stimuli. For example, when some kind of infection is produced, it needs this type of immunoglobulin, so the gene will be rearranged to produce that type. So this is another method of gene expression at the DNA level, even before the transcription is taking place. So next thing is transposons and gene switching. So what is transposons? It is also known as jumping genes. So what happens here? A DNA sequence, it moves to a new genomic position. Normally it will be present here in the gene sequence. So it moves from here to this position and producing a new sequence. So what happens when transcription is taking place? It will produce another type of protein. So the desired protein is present here. If this sequence is present in more amount of transcription of that region will take place. So how this jumping is taking place? By the help of transposase active, and this was discovered by Barbara McClintock, and this revolutionized the understanding of genome dynamics. That is, our genetic material or gene sequence is very dynamic due to the presence of this jumping genes. So a DNA sequence present here can jump to another area through the transposase activity. And what is retroposons? That is, here the DNA jumps to other region, but with the help of RNA. It will be converted to RNA, and that will be again changed to DNA by reverse transcription process. It is nothing but a jumping gene process, but here the DNA is first converted to RNA by transcription, and then reverse transcription, it is converted again back to the DNA at the other position where it is going to jump. So that is known as retroposon. So the other two things are gene switching and gene silencing. That is, when a particular product is needed, that gene will be switched on, and when that particular product is not needed, that gene will be switched off. That is, gene switching. And this off position, that is also known as gene silencing. So how this gene silencing is taking place? It can occur at two ways. One is epigenetic mechanism, that is at the DNA level by modifying the histone or the methylation of the DNA. And other thing is RNA interference, that is what we have seen as the mRNA or siRNA, small interference RNA. This sequence present at the transcript will promote their degradation, and that will result in gene silencing. So even though transcription is taking place, it will not be producing protein, because at the mRNA level itself, due to the presence of this interference RNA, that is siRNA or miRNA, that will promote the degradation of mRNA. So that gene will not be expressed. So that is known as gene silencing. So here we are studying various terms like jumping genes, retroposons movement or gene switching or gene silencing. So these are various other possible mechanisms by the gene expression is controlled. So the gene expression is controlled in enormous way, various ways, like at the level of transcription, at the DNA. So we are studying various concepts of regulation of gene expression. So next thing is epigenetics. So this is another form of regulation of gene expression at the level of DNA. So what is epigenetics? It is above genetics. That means when we alter a gene, or what we seen in the gene amplification or uh, gene rearrangement, what is happening here? There we are altering the sequence, or we are producing more number of sequences. But epigenetics is nothing to do with the nucleotide sequence. It will not alter the DNA nucleotide sequence. It will do only modifications in the chromatin structure. That is, it will either methylate the DNA or it will do some modification at the histone modifications. So that is known as epigenetic modification. It is above genetics. No DNA nucleotide sequence is changed in the epigenetic modification. So there will not be any mutation. When you are going to change a nucleotide sequence, it will result in mutation. So no mutation in epigenetic modification. So the advantage of this is it is a dynamic regulation. What the change we do is a reversible modification. So that allows a flexible gene expression control mechanism. Whereas if you change a nucleotide sequence, that will become that will not be a dynamic or a reversible modification. That will result in producing a wrong protein or mutated protein. So that is the advantage of epigenetics. That is, it is a dynamic regulation, it is a reversible method, and it gives a flexible gene expression control mechanism. So this is the picture which help you to understand what is epigenetics. So basically this is what the DNA at the end. But how this DNA is organized in the chromosome, it will be wound on the histone protein, and it will be tightly packed, and it will be condensed to a chromosome, and this is the final chromosome. So this is what we see in the chromosome when we magnify the chromosome progressively. So what we are doing in the epigenetics, we are doing some modifications here. For example, if you methylate this structure, this winding or this coiling will become more. So more amount of this DNA will be coiled and tightly packed. So less amount of DNA will be available for expression or transcription. So increased methylation causes more tightly packed structure of chromatin and reduces the expression. So likewise, we are doing some chemical modification here. We are not at all touching this nuclear or nitrogenous bases present in the DNA sequence. No sequence moderation. Only chemical changes here, and by that we are controlling the expression. That is known as epigenetic expression. Epigenetic regulation of gene expression. So first epigenetic modification is methylation, DNA methylation. So the DNA has certain area known as CpG islands, that is cytosine rich area, and this CpG island is more prone for methylation, and the enzyme helping for this methylation is methyltransferase. So when methylation is increased, as we have seen previously, when more amount of DNA methylation is taking place, there is tightly packing of the DNA in the chromatin structure. So less amount of expression will be there. That is known as gene silencing. And when we need to increase the transcription rate, we need to demethylate. So the CpG island, that is more prone for methylation, will be present in promoters. So by doing methylation in the CpG island in the promoter, that we can control the transcription initiation. So this methylation, here we are not at all altering the nitrogenous bases or nitrogenous sequences. Only we are methylating, and that is more prone at the cytosine rich region, that is CpG island. And if you target that island in the promoter region, by increasing the methylation, we can reduce the transcription. So that is a concept of DNA methylation. So another epigenetic modification is histone modification. That is, histone is the protein which is help in the tightly packing nature of the DNA in the chromatid. So if you acetylate the histone, that is histone acetylation will increase the transcription. So by doing this acetylation reaction, we can control the transcription. Either we can increase by more acetylation. If we deacetylate, it will decrease the gene transcription. And these are the other modification which we can do at the histone, and that modification will alter the gene expression by these modalities. So there are some diseases in our body which are occurring due to this epigenetic modification. So if there is some defect in this epigenetic or there's some modification happening in this epigenetic level, that also can lead to some diseases. One example is Fragile X syndrome. So the gene responsible for this is FMR1 gene. So more amount of methylation is taking place at the promoter region, and that results in gene silencing. So the gene is not expressing. So the FMR1 gene is not expressing, that results in Fragile X syndrome. So another thing is cancer. So in cancer, we know there are two types of genes commonly. That is, one is proto-oncogene that will promote the cancer, and another thing is tumor suppressor gene. So that will inhibit the cancer. So it will act as a brake. It will stop the rapidly proliferating cancer to slow down. So this is the role of the protein produced from the tumor suppressor gene. So what happens in cancer? This brake need to be in a failure state. So the brake should not be functioning. So what happens in cancer? This tumor suppressor gene will be methylated. So the hypermethylation of tumor suppressor gene will reduce or silence the tumor suppressor gene. So there will not be any break. So the cancer can rapidly proliferate. So that is what happened in cancer epigenetics. So an epigenetic modification in the tumor suppressor gene will lead to the progression of the cancer. So next thing is genomic imprinting. That means a particular chromosome 15, in that one for example, in the chromosome we have both the components, paternal and maternal. So one will be imprinted, so other will not be inherited. So that leads to certain disorders like Prader-Willi syndrome and Angelman syndromes. So knowing this changes will cause certain diseases, like some epigenetic modification is causing cancer, will help us to design certain drugs to target this epigenetic level of modification. So that is DNA methyltransferase. That is an enzyme responsible for this hypermethylation. So if we target that enzyme, we can reduce the progression of cancer. So we can some drugs are produced based on this targets, like azacitidine, decitabine, vornostat. So these treat cancers. These are the drugs which used to treat cancers by reducing this epigenetic abnormality, thereby reducing the cancer burden. There is a term called mitochondrial DNA. So usually DNA will be produced in the nucleus. So only the mitochondria has its own DNA. It has a gene content of 37 structural genes, including 2 rRNA and 22 tRNA and 13 proteins. So this also encodes the subunits for the electron transport chain, like NADH dehydrogenase, cytochrome oxidase, ATP synthase. So these are encoded from this mitochondrial DNA, and it has a unique code. For example, UGC or certain stop codons present in normal genetic material will become a coding for certain amino acids in the mitochondrial DNA structure, and it will follow the cytoplasmic inheritance pattern. To summarize, we have seen what is gene regulation and why gene regulation is needed for our body, and what are the various levels at which the gene regulation can take place. The most important level is transcription level. and other thing is DNA level. And we have seen about epigenetic modification. So this also contribute for gene regulation. And we have seen a few words about mitochondrial DNA and how the at the level of transcription regulation is taking place. We used a model lag operon to understand this, and also this altering the stability of mRNA also help in the regulation of gene expression. In DNA level, by gene amplification or gene rearrangement, we can control the expression, and jumping genes, that is transposons, and gene switching mechanisms can also result in regulation of gene expression. So under epigenetics, we have seen two major types of chemical modification. One is methylation. So the common site is CGP island, and the other is histone modification. So that is a histone acetylation reaction is the most common important reaction. So these are the things which we have seen under regulation of gene expression in our body. So also we have seen some disorders happening due to the epigenetic modification. That is, cancer is happening due to increased methylation that causes decreased expression of tumor suppressor genes. Likewise, we have seen some disorders related to epigenetics. Thank you.